Andrea worked with Rod in Arizona for several years, and they published some big stuff together. It was very impressive, actually. And he also has been, you did your PhD with Michele Morgante?
Yes, I did.
Right?
Yes,
and on transposons,
yes.
which is also the topic of today's seminar. And Andrea came to us after 7 years, I think, in Pisa at the Scuola Superiore, Sant'Anna. And apologies for my pronunciation just then.
No, it's
and
good, I would say. Yes, it's good.
And Andrea is here as a pretty important guy in the center. Bioinformatics is, in many ways, I think a lot of you know my feelings about this, is extremely important component of the future of plant science, and that interface between math, computing, and biology, I think, is an extremely important area for making breakthroughs. And this is exactly what Andrea is in. He's also a drop dead nice guy and incredibly collaborative and supportive. So it's a pleasure to have you here, Andrea, and it's a pleasure to, by proxy for Rod, be able to have the chance to speak for 2 minutes and introduce you. I'll leave the floor to you now, sir.
Okay. Thanks a lot, Mark, for a really nice introduction. And
completely spontaneous.
Yes, indeed, I appreciate that a lot. And thanks to all of you, obviously, for attending this seminar. Today, I would like to talk about the transposable elements in plants. And my focus usually is plant comparative genomics. And so I will undertake a comparative approach to this important component of the genomes. And basically today I will provide a very, very brief introduction to transposable elements. Then I will start comparing their features and evolutionary dynamics in two very different and distant plants: an angiosperm, that is the domestic rice, and the gymnosperm, that is the Norway spruce, Picea abies, the Christmas tree, to make it clear. From there, I will move to a smaller and well-described system covering a much shorter evolutionary time span, that is the Oryza genus. And then I will zoom in to T dynamics at intraspecies level, focusing on Oryza sativa accessions. And then I will finish up describing, introducing a recently funded research project here at KAUST, dealing with exploring the genetic variability originated by teas and how we can think of use it to improve the tolerance to drought in domestic rice. TEs are sequences of DNA that can move around the genome from one position to another by means of a process called transposition. There are many different types of TEs, but they can be classified in two major classes. Class 1, also called retrotransposons. They are capable they move through an RNA intermediate. They are transcribed and then retrotranscribed elsewhere and inserted elsewhere in the genome. If they were a text editor, we can describe their movement as a copy and paste action. And then we have class 2 elements, also called DNA Ts, and they just move from one position to the other inside the host genome. They are excised and moved elsewhere by an enzyme called transposase. If they were a text editor, their movement would be similar to a cut and paste action. For many years, Ts have been dismissed as junk DNA, and in the best scenario, they were considered a sort of an oddity. And then during the years, evidence accumulated demonstrating instead that they are important player in functioning and evolution of the genomes. And here are listed some of the most important features. They are, as you can imagine, mutagenic. They can insert into a gene, inactivating it. They can affect transcription, and they are also involved in many genome rearrangements. Okay, In these pictures, you can see some of the most spectacular effects of TE on plant phenotypes, starting from the iconic spotted kernel of maize to the pulp of the color of the pulp in oranges. Even the color of the skin in grape berries is due to TEs, and also the color of date is controlled somehow by TEs. Not just that, they constitute a significant portion of many eukaryotic genomes, up to 85% of the maize one. So they are a major force behind the genome size variation. They can grab, duplicate, move, reshuffle the part of genes. They are valuable biotechnological tools. We can use them to inactivate genes, and we can use them in functional genomics studies. They also contribute to the evolution of new genes. When I started my PhD at the time, the sequencing of the domestic rice genome was in full swing, and a lot of sequences were available. I also was in a lab that has many interests, but has also a strong research line focusing on conifer genomes. And so it was quite natural for me to carry out a comparison between these two different genomes. I'm talking of the genome of Norway spruce and rice. The two species are definitely far apart. From an evolutionary point of view, there are more than 300 million years of evolution separating them. Norway spruce has a huge genome, 20 gigabases, that is almost 7 times larger than the human genome, whereas rice has a compact genome, 390 megabases. The amount of genes, as you can expect, is comparable in these two plants, 28,000 in spruce, 36,000 in rice. Obviously, the genome size difference is not due to genes. Both of them are diploid, and this is just by chance, the amount of chromosome per haploid complement in both cases is 12. These striking differences obviously puzzled scientists for years, and one of the very first thing we checked was the T content in the two plants. And not surprisingly, both the relative and the absolute amount of TEs is much greater in Norway spruce than in rice. However, all the major types of these elements are present in both genomes. Once more, pointing out that TEs are quite old from an evolutionary point of view, since they are ubiquitous in many organisms, in all the organisms, and so they have to be quite old. Things started to become quite different when we took into consideration the dynamics of the TE amplification in the two groups. In the case of spruce, but more generally in the case of conifers, the extant TE population derived from events that accumulated over 10 million years. Here in this graph on the x-axis, we have the time. On the y-axis, we have the amount of TE that were inserted in that time. In the case of rice, but I would say most of the crops, actually the extant TE population is quite young and accumulated just over a few million years of evolution. In this graph, actually the time is on the y-axis. Okay, so things were quite recent for rice. Not just that, if we focus on a really recent evolutionary time, and I mean anything less than 4 or 5 million years ago, then we can see that there is plenty of evidence of TE activity in rice, here in yellow, in this histogram, whereas basically there is no activity in the case of spruce. The host genome can counteract the amplification of these, trying to remove them. There are different mechanisms to do that. One of the most effective is unequal recombination. Very, very briefly, here I present the canonical structure of a non-terminal repeat retroelement. These two regions, called LTRs, are identical or highly similar when the element insert. and they can trigger a recombination, during which one of the two is removed, as well as all the intervening region between the two, leaving behind a so-called solo LTR. Now, the intensity of this process can be somehow evaluated, figuring out the ratio between solo LTRs and complete elements. In the case of Spruce, we have on average 5 complete elements per a single solo LTR, whereas in the case of rice, we have 1.52 solo LTRs per complete element. And so the removal of these elements is much more effective in rice. But how fast this removal take place? That is, how long will a TE remain, will be retaining the host genome on average? In this picture is depicted an orthologous region shared between domestic rice, Oryza sativa, the African rice, Oryza glaberrima, and a wild rice, Oryza brachyantha. Genes are depicted in black, Ts in blue and red. And the time that separates sativa from brachyantha is 9 million years. What we can see is that most of the genes are actually retained in the orthologous position as expected, but when it comes to Ts, very few are, some of them are retained between sativa and glaberrima that are close enough. No one of them is retained in the same position between sativa and brachyantha. They have already been removed. And indeed, the removal, the half-life of TE in rice is around 3 million years, but we will come back to this later. What happened in spruce, here is a comparison between white spruce, Picea glauca, and the Norway spruce. They separated 13 to 20 million years ago, according to different estimates. Well, in this case, out of 120 LTRRTs identified in Picea glauca, more than half, 63, can be identified in orthologous position in Norway spruce. That means that they are still retained after 13 to 20 million years. To summarize all of this, in rice we have a small, absolute and relative amount of T. We have evidence of recent and sustained activity. There is a fast turnover of these elements and an effective removal of these elements. Whereas in spruce, we have an enormous amount of Ts. They are ancient, no evidence of present activity, slow Ts turnover, and limited removal. So thinking of a genome as a bucket and Ts as a water, this is the scenario in rice, where we have a lot of water pouring in the genome, but promptly removed. Whereas in the case of spruce, there is a drop falling into the bucket here and there during the time, but with a very inefficient removal. And so, thinking of all of this, we were able to provide a possible explanation for the huge size of the spruce genome. And we said that it seems to result from the slow and steady accumulation of a diverse set of Ts, and also it is due to the lacking of an efficient elimination mechanism there. Okay, but what about the causal mechanism of all of this? It remains still unclear, but during the years we collected some information and we could at least partially explain what is going on there. And the first thing is that by means of comparative analysis, we were able to prove that the reduced T removal mostly is related to genome size. That is, the largest the genome, the less effective is the removal, as if once a certain obesity genome threshold is reached, then the genome fall in a state of genome paralysis, basically. Not just that, but for Norway spruce, we were able to prove that out of many plant genomes, it is one of the most heavily methylated in all the context, and especially in the regions, as expected, that are related to Ts. And so, altogether, basically, the low recombination frequency would more likely result from hypermethylation. Okay, so comparison of the dynamics across large evolutionary times pointed out striking differences in amplification and removal of these elements in angiosperms and gymnosperms, suggested a viable model for the conifer genome evolution, and allowed at least a partial exploration of a possible causal mechanism behind those differences. Now, all of this is quite interesting from a biological point of view, and but is somehow expected because indeed we were comparing two very different organisms. Also, it is quite likely that in doing so, we were missing some important piece of information, many details for sure, and so it would be interesting to focus on the comparison in a somehow more limited evolutionary time frame. And in this sense, the Oryza genus provides a wonderful system. It includes 27 species that are arranged in 11 different distinct genome types. And it includes 2 species that are domesticated, the Asian rice, Oryza sativa, the African rice, Oryza glaberrima. The evolutionary history of the genus spans 15 million years of evolution. These species actually cover a wide range of different habitats, and also, most interestingly, a significant variation in genome size can be appreciated inside this genus, going from the smallest genome, 357 megabases for Oryza glaberrima, to the largest one, 1283 megabases for Oryza ridley. Not just that, but for Oryza, it's available a huge amount of very valuable genomics data that were produced during the years in the frame of some ambitious research efforts set up and coordinated by Professor Wing. And I'm talking of OMAP, Oryza Map Alignment Project, that then became IOMAP, International OMAP, because many groups from around the world joined these efforts, and also OGI, Oryza Genome Evolution. The idea behind these efforts was to try to unlock the amount of variability present in the genus by means of comparative genomics study that would have provided data then to study genome organization, evolution, polyploidization, development, transposable elements, and so on and so forth. Since the very beginning, we paid attention to transposable elements. At the time, we were using mostly random shear libraries that provide a shallow coverage of the host genomes. Reads were obtained using Sanger technology, but in any cases, they provide really useful information. Here are the results for an analysis of 12 different genomes listed here. And in analyzing them, we were able to prove that all the T types were present in all the species we consider across the genus, that the most abundant of these types is that of long-terminal repeat retroelements. And also, we were able to appreciate how the abundance of these Ts were different in different species. Here, we went into detail regarding 26 families of long-terminal repeat retroelements, 11 copia and 15 GIPs that were searched across the 12 species. And it is possible to appreciate 2 important things. First, that all of these families are represented in each single species. So there is no family that is exclusive of one or few species. But what is quite different is the amount of copies that the very same family was capable to attain in different genomes. Here, the arrows pointed out 3 examples: Atlantis, here, Rayar 2, there, and Rayar 1. Possibly the most spectacular and famous case of this differential amplification is that of Oryza australiensis, that was described by the group of Professor Olivier Panot. And basically, they were able to detail the explosive amplification of just 3 families of teas named Ryer1, Kangaroo, and Wallaby, that alone were able to provide more than 60% of the genome size of these wild Oryza species. And all of these happened during a very limited amount of time, an evolutionary point of view, 3 million years, with 2 amplification peaks around 1 and 1.5 million years ago for 2 of these species. In this southern blot, you can see that that was done using probes from these 3 elements, that they are somehow present across all the species of the genus, but they are mostly abundant indeed in Oryza australiensis. The insertion time estimates and the rapid amplification of these elements was also confirmed by the phylogenetic analysis in these 3 trees. The branches in red refers to Oryza australiensis paralogs. And basically, the authors very nicely and effectively summarize what they have seen and described in the title of this paper that says doubling genome size without polyploidization. And indeed, this is what happened in Oryza australiensis. We studied some similar dynamics in the most basal Oryza species, Oryza granulata, where a single family named Gran3 amplified up to 20,000 copies, 10,000 of copies actually, altogether making up 22% of the genome. And all of these occurred over just 4 or 5 million years of evolution in 3 waves of amplification. In this fish image, you can see in light green the probes from Gran3 scattered and evenly distributed around the Oryza glaberrima chromosomes. Another family, TY3 Atlantis, this didn't attain the high amount of other examples, but still is one of the major components of many genomes, especially in the officinalis complex. The officinalis complex include Oryza puntata, officinalis minuta alta australiensis, as well as in Oryza granulata and Ridley. What is interesting about Atlantis is that it was capable to amplify successfully in more than one host gene. Now, these are the major driving force behind genome size variation in the genus Oryza. All the T families are shared across the genus, but had different fates in different genomic backgrounds. And the T amplification occurred over short and somehow recent evolutionary times. Later on, we had the possibility to deal with entire genome assemblies. Here are listed some of them, and along with the species is specified which group took the lead in the genome sequencing and assembly. Along with this, there are other genomes that we are currently sequencing and assembling, and basically by the end of this year, we should have an assembly for each of the Oryza species, with the exception possibly of Oryza neocaledonica that will follow. We have also the genome sequence for the outgroup Liersia perieri. It is important to point out that many of these assemblies actually are of very high quality because they were obtained with a PacBio technology supported by optical maps. And so the final assembly in many cases is GAP3, contiguous, of very high quality across its entire length. So basically we are dealing with platinum standard genomes. The first complete genome available beside Oryza sativa was that of the African rice, Oryza glaberrima, that has been obtained by a classical hierarchical approach. That means back library, physical map, identification of a minimum tiling path, and then sequencing of each back clone. The sequencing was done with Sanger and at the time also 454. The genome size of this species is 357 megabases. It contains many important adaptive traits for cultivation. And what is interesting, especially for me, is the evolutionary distance from Oryza sativa, just 0.6, 1.2 million years. As far as these are concerned, the amount of these elements is greater in domestic rice than in the African rice. In African rice, the total amount is 104 megabases, 156 in the Asian rice. The difference, 52 megabases, is pretty close to the difference in genome size. And that means that the Ts alone can explain the difference in genome size between these two closely related species. We study the distance of Ts from predicted genes there, and out of more than 28,000 genes, more than half of them has the closest T at less than 1 kb. 63% of these Ts are mites that are so enriched close to the genes. 16% are long-terminal repeat retroelements that are depleted there. A similar trend holds true also for the more than 16,000 genes that has an insertion of T fragments inside. 60% of them as a mite, or more than one mite. 16% have an LTR retroelements. Only in 400 exons we had an insertion of Ts. Then we studied the insertion. We studied the insertion of these elements in the two genomes, trying to figure out which of them occurred before the speciation of Oryza glaberrima and Oryza sativa. So how many inserted in the last common ancestor between the two. Basically, that happened for 54.2% of the Ts we consider. How many of them inserted just in the Oryza glaberrima lineage here, and how many in the Oryza sativa 1? In the case of glaberrima, 11.5%. In the case of sativa, 34.4%. And basically, it is confirmed that the recent transpositional activity have been greater in the domestic rice lineage. Later on, not just Oryza glaberrima, but many other complete assemblies were available, and that provided the opportunity to go into detail and carry out more comprehensive analysis of these in this system. The very first thing we did was to check the abundance of this element, and indeed we proved that they are definitely quite abundant across the different Oryza species, but there is a lot of variability in this abundance, and basically the content of repeats including Ts goes from 27 to 50%. And once more, we were able to confirm that each species was distinguished from the other by the different amount of long-terminal repeat retroelements for different families. Selective amplification of Ts has a key role in defining chromosome shape and evolution. And we studied this kind of events using phylogenetic analysis across the entire genus. Here are presented two trees, one referring to TY1 copia, the other two referring to gypsy elements. And analyzing them, we were able to spot some lineage-specific transpositional bursts. Like in this case, all these green branches refer to the amplification of the Atlantis element in Oryza punctata. Not just that, we were also able, using the entire genome sequence, to figuring out the insertion time of these elements, and altogether, basically, We were able to identify several lineage-specific transpositional bursts, most of which occur in the last 2.5 million years. And the recent species-specific amplification bursts for LTRRT population explains their different abundance in different species. We were also able to study the removal of these elements. We analyzed 75 orthologous LTRRT loci, and for each of these locus, we figure out how many of the T sequence was removed. We figure out that comparing the remnants to the original complete element. And then we averaged the total amount of sequence removed to the radiation time that originated this species. In doing that, we figure out that the removal rate is 3.62 kb per million year per element. This number in itself doesn't tell you too much, but think of it in this way. Basically, it's telling us that approximately 1/4 of the rice genome will be eliminated in the next 3 to 4 million years, unless new waves of transposition will take place during the same period. Most of the Ts are removed, but some of them are fixed and retained. Here is an example of this element that is being retained in the intron 4 of this gene that is present across the entire genus and beyond, because the gene and the insertion is present also in the outgroup Liersia perieri. The analysis of the OMAP data allow us to get further insights in the role played by Ts in genome size variation. We clarified tempo, mode, subjects, and in genome sequence variation, we were able to identify presence, absence, polymorphism, and species-specific proliferation events. We were able to see how the fast turnover of these elements in Oryza is due to recent specific lineage-specific amplification and a high deletion rate. We have seen that the families are shared genus-wide, but their insertions are polymorphic, and this insertional polymorphism can be exploited for breeding in addition to the diversity that we found in genes themselves. During the years, several attempts to study the genomic variation of domestic rice were carried out. One of the most successful resequenced 3,000 accessions using Illumina. The reads were then mapped back onto the reference that was the variety Nippon Bare for Oryza sativa, identifying structural variants, SNPs, and indels, and using this information to infer the phylogeny of all the subpopulation of rice. Authors at the time were able to identify 9 subpopulations, but other studies then added 6 more subpopulations that are depicted here. This effort was great and provided very, very useful data, but suffers from one limitation, that is that only one reference genome was used. And so what if a subpopulation-specific region is available? That would have been missed. So in order to fill this gap, we tried to provide reference genome assemblies for all the subpopulations for which these sequences were not available, basically 12 of these subpopulations. And the effort was mostly carried out by Dr. Yong Zhu, who is a postdoc in Professor Wing's lab. Accessions were sequenced using PacBio. They were assembled also using optical maps, and the final result was heavily manually edited using a tool named Genome Puzzle Master, a tool that has been developed in Professor Wing's lab. And the final result of all these efforts is a data set of platinum standard genomes that makes altogether a wonderful set of data for many kind of analysis, but in my case, especially for the identification of structural variants. Structural variants are defined as genomic alterations longer than 50 base pairs. We can have deletions, insertions, duplications, translocations, inversions. And we carried out pairwise comparison between all the different sequences we have for these subpopulations. In doing that, we were able to identify, on average, tens of thousands of structural variants for each comparison. And these tens of thousands of variants translate into a total genome sequence on average of 33 megabases of sequence that is not shared between any of the two accessions compared. That, by the way, corresponded to roughly 8% of the genome size of Oryza. We studied the length distribution of these structural variants, and the distribution is skewed towards short, let us call them short sequences around 500 base pairs. This length is consistent with the length of many TE elements, such as mites, trim, sol LTR, and TE remnants. Indeed, when we analyzed these sequences, we were able to see that SVs were enriched in TEs. Indeed, 85% of them, of their sequence, is represented by TEs, whereas the average content of TEs across the entire genome here is, in this graph, is depicted by these blue bars, is just less than 50%. So SV are definitely enriched in TEs. We checked the distance of structural variants from the nearby genes. Here is the comparison of the accession Nippon-Bare versus Chaomeo. 12,000 SVs were considered, and 1 out of 5 is basically inserted into a gene or located less than 1 kb upstream from the nearest gene. In particular, 13% is less than 1 kb upstream, and 8.64% of the structural variants are inside genes. That is, in this case, 1,000 of them, of which 467 are within exons and 577 in introns. We then started try to see if all the Ts were involved in structural variants generation or just a few of them. We carried out phylogenetic analysis. This refers to non-terminal repeat retroelements. Here in red are depicted paralogs from the most abundant T classes in domestic rice, and they are named here. And very simple and briefly, what we can see in this image is that all the families are involved in the creation of structural variants. So the entire complement of Ts is involved, not just a few families. I think I can skip this slide, and basically comparing 16 genomes, we were able to identify the tens of thousands of structural variants with an average amount of 33.7 megabases per genome. The variability was greater between accessions that belongs to far apart subgroups, as you can expect. The overall majority of SVs originated from T activity, mostly long-terminal repeat retroelements and mites. A significant fraction of the SVs is located in close proximity, less than 1 kb upstream of known genes, and so very likely they can contribute to phenotypic variability. Now, the fact that stress heavily affects T activity is well known since the very beginning, since the first seminal studies by Barbara McLean. And however, the pattern of activation and activity can be somehow complex. Here are some examples. Stress, here Ts are depicted in red, genes in black. Stress can activate a gene, like in this case, T, like in this case, then the T got inactivated. Or here we have two different Ts. The stress activate one of the two, repressed the other one. Or an already active Ts can be repressed actually by stress and later on can be activated. What is less known is how and if T insertional variability can lead to stress tolerance resistant in plants. What is the idea behind of this question? Well, these are major source of natural variation. They have an important role in gene regulation. Stress can activate them, and now new T insertion could confer stress responsiveness to neighboring genes and can contribute to stress response regulatory networks. It is a fictional example. We have a stress that trigger T activity, these purple triangles, in this population of plants. And one of the possible insertional patterns actually lead to some tolerance resistance, this plant. And then the selection quite likely will positively select for this plant, and this pattern of insertion will be fixed across the population. Now we want to study that in a interesting system and dealing with an important issue. So we decide to tackle drought in rice, and we propose a grant that was written by Professor Wing, and KAUST funded that grant. Also, from the research point of view, other people are involved, in particular Dr. Amelia Henry, who is a plant physiologist at IRRI, Philippines. He has a strong background and knowledge of drought resistance in rice. And also myself, I am involved in this research. First step would be the identification of 160 rice accessions representative of all the 15 subpopulation. We will maximize the genetic diversity between them, and we will also try to select accession with different tolerance to drought stress. All of them will be resequenced using PacBio hi-fi reads, and the data will be used to define SBs between them. Then each accession will undergo 3 watering regimes for 2 seasons. We will have a control, well watered, then intermittent drought stress, and progressive drought stress. The accession will be phenotype for drought tolerance and agronomic performance during these processes, and they will be also characterized from the transcriptome point of view in order to identify differential expressed genes and figuring out the partition of expression due to genotype, treatment, and genotype by treatment. Then, finally, we will use all SBs, phenotypic data, transcriptome data, genomic data to identify the EQTLs, okay? And them will indicate likely cis-regulatory variants that affect the steady state gene expression level in different genotypes and their perturbation under 2 specific water regimes. We will then focus on those variants having the most of these cis-regulatory variants. We will create F1 progeny from them, and we will go into detail studying what is going on this progeny. We will take care of allele-specific analysis. We will carry out total RNA analysis, and also we will analyze methylation there. So the idea is that we should, being able in doing so to unravel the large but poorly exploited reserve of natural variability represented by structural variants, we will hopefully disentangle the whole genome scale relationship between Ts and drought stress response. We will identify a large number of molecular markers that can be later on effectively used for improved drought tolerance in rice. Data, we will set the stage for functional genomic studies aimed at the identification and exploitation of molecular mechanisms. And it's worth to be noted that we will analyze different kind of drought stress. And so this possibly will allow us to identify genes and mechanisms related to fast drought recovery. And this would be a novel trait. So to conclude, basically today we move from a comparison between very different taxonomic groups, two intraspecies comparison. We have seen how different evolutionary timescale provided different opportunities in studying and disentangling T activity and dynamics. Comparative platforms as IOMAP and OG provided a comprehensive set of knowledge and genomics tools for crop improvement. In Oryza, platinum standard genomes for each of the subpopulation of sativa can be used to characterize clearly the natural standing variation within Asian cultivated rice, and will also provide accurate sequence data for further analysis of TEs, centromeric regions, telomeric regions, structural variants, gene annotation. There is one thing that I want to point out at the end is I want to quote this sentence from Viemont Vieira that has been published in a commentary in Nature many years ago. At this point, 15 years ago, they said, what was once dismissed as junk DNA must now be regarded as a major player in many of the processes that shape the genome. Okay, things are starting to change, but it seems to me that we are still lagging a little bit behind in figuring out completely the meaning of this sentence. But admittedly, I am quite biased. However, consider that. And obviously, there is a huge amount of people that we have to say thank to them and acknowledge their efforts, all the IOMAP and OG collaborators listed here, all the people at the Arizona Genomics Institute listed here, the Wing Lab at KAUST, and clearly all the founding agencies that are listed here and KAUST too. Thank you. Now, I am open to answer your questions. Okay. I have a few questions here. The first one is how you define stress. Well, again, I am not a plant physiologist. I am not an agronomist. However, I define stress and as any reaction that is significantly different from a steady state from what you expect plant behave. And so, we will take care of, we will analyze several different parameters there. But again, I'm interested in basically to see how the population we are dealing with will cope with the stress. Basically, how long they survive, how much they will provide as a yield, and eventually how much they will recover after the stress and things like that. But admittedly, things are much more complicated than that. Okay. And okay, then I have a question from Saul, and okay, basically she's asking which are the general triggers from such waves. Well, obviously we can just speculate about those, but I would say that, as I pointed out later on, that definitely any kind of stress can be a quite effective trigger of this wave of retrotransposition. And also, well, in the case of polyploids, the polyploidization itself could be more than enough to trigger a response. But, and the stress, you can think of climate changes and or the reaction to a pathogen and things like that. There are many things that can perturbate the steady state of the host genome and triggering the activity of these Ts. And then Robert Lehman is asking if my experience are T expansion events usually driven by a single T or large groups of Ts. I would say that usually, at least the most spectacular and impressive and fast expansions usually involve just one family, in most of the cases. Yes. And then I have Young. Okay, please, Young.
Yes, Andrea.
Yes.
Oh, sorry. I'm waiting for my camera. Okay. Hi, Andrea. Thanks for your great talk, and it's very interesting and bring a lot of ideas. And I have two questions. Basically, the first one is in your experience, and no matter it's in the Oryza, sativa, or Oryza genus, or trace, and the T is expansion that or turnover that like you think that is randomly or basically from the different T families structure or sequence similarities expansion or turnover system, or they are just randomly changed and will have effect on the phenotype, no matter it's tolerance or salt tolerance or heat or drought tolerance or
well, I mean, these kind of things, I'm curious actually, because what I expect from that approach is at the very beginning, a bunch of correlations, then will have to be validated functionally. Okay, and regarding this correlation, what I expect to see is that probably we will have some single elements that are capable to have an effect, but I am also curious to see what happened looking at all of them together, especially certain classes such as DNA Ts, because they can create regulatory networks that can have an effect on what we see. And this is incredibly complex, but possibly is one of the most interesting things that I would love to study there.
Yeah, not only the different type of families, but also different family or subfamilies of the also we can call that different style of one element, right? But that also compare with the genes evolutions histories that the genes evolution histories are always, as far as I know, it's always like a mess, and it will be quite important and also interesting to figure out that will be huge discoveries in no matter it's plants or animals. That will be very interesting. And another thing is, as you just also mentioned, I noticed on my notes, and if that is only just one family or one type of element could be needing to be expansions or turnovers. And if we started like that, if they have no, I mean this question maybe you keep that open, or maybe you already have some ideas. If that is could be the genes variations, could be the T's movement, could be leading by some environment, by some pressure from outside, or for example some environment, some cold temperature like that. If in that case, I don't know whether this is an open question or already not have answers. Then when we study like specific phenotypes, we will find some strong relationship with some specific T's, but that doesn't match the genome's expansion.
Oh, yes. I mean, not necessarily the most effective TEs are the most abundant ones. I do agree. I do agree.
I don't know about that. But I was just thinking if it could be leading by some stress phenotypings, then maybe when we study that, we will be only find the results quite relationship, quite high functional regarding to that family or that specific element. But I do really don't have any idea about that.
We'll see.
Yes, thank you again for the great topic. It's very, very interesting.
Thank you. And then, I mean, Hanin, it seems to me, she raised her hand for a question.
Yes. Hi, thank you so much for your talk. And as I said before in the morning, you're a really great teacher.
Thank you.
Yes, thank you. I have two questions. One is, so you showed that in the different rice species that they have the same LTR families, but
Yes.
the difference is in the abundance of these families.
Yes, mostly, yes.
Yeah, so these are between the diploid rice species, the different diploid rice species, right?
Yes, go.
Yes. So what I wanted to ask is, how about the polyploid rice species? So, because like, for example, in wheat, or bread wheat, hexaploid wheat, but they found that that there was difference in the abundance in the subfamily level, but not the family level. So I wonder if between the rice is something like this, that is the difference still in families or subfamilies level?
Well, I mean, I remember that when I was dealing with polyploids, I was a little bit annoyed, right? And because I didn't find any major expansion of these elements as I expected. As I mean, we expected to see a lot of them. Instead, the amount was pretty reasonable. And again, I would also most of the families, usually most of the paralogs of the very same families tend to mix together. So their amplification was quite recent.
Mm-hmm.
And so, no, I didn't saw any particular difference between families and subfamilies.
But in the tetraploid,
yes.
Okay.
Yes. And
okay.
They were quite mixed, and I mean, there was nothing specific for one of the two counterparts.
Mm-hmm.
Let us put it this way, yes.
Okay, and another thing is, it might be just a comment.
Okay.
Yes, for the last project or the funded
yes.
project that you will work in is the tandem. So the tandem duplication is actually, I mean, as you know, that it involved in plant adaptation. And it's a very nice thing that what you want to do is to look for structural variants, and then you will do also RNA gene expression analysis, because it would be very nice to, in particular, look at how the tandem duplication can actually affect the gene expression, in particular, the one that involve in the adaptation.
Yes, I mean, as you have seen, we were talking of mostly of structural variants that clearly involve Ts, but are not limited to Ts. And so from this point of view, also the data we are going to provide that can be used in many different ways. I mean, obviously I focus on what I was interested in, but actually I think that will be plenty of information for
that you can get,
looking for answers for many different questions. And as I said, but I repeat that once more, clearly the most obvious outcome would be a huge amount of correlations. But then that will open, I mean, the problem, okay, how we can validate them and move from a correlation to causation. And I'm quite excited, actually, looking for this data.
Yeah, it's actually very nice. But okay, again, back to the polyploid rice.
Okay.
I think, okay, I think maybe I did not ask it correctly, or I misunderstand you. What I meant is about not the difference between the polyploid rice, two different polyploid rice species,
yes, but
the subgenome.
Yes.
Okay, so there is no actually difference in the abundance of subfamilies or okay.
No. later on, well, I mean, I don't know if we have started to see something like that in Oryza Ridley, but we are at the very beginning.
Ah, okay, okay.
That's why I don't want to comment too much about that. But
Okay.
there, possibly that would be the first example of striking differences. So far, let us say minuta, even cortata, and alta doesn't seem to be that.
Yeah. Okay.
But you have to be patient for a few months.
Okay. Thank you. Thank you so much.
And then I have
there are another two hands up, but I see a silence, so I want to follow in the Hani's question that regarding to that T's and the large structure variance is pretty hot in the genome these years, and so the basic consent is about to identify the not about any drought or heat tolerance responding from large structure variations. And then it could be affect about the 3D or insert the promoters or genes insertions to enhance or lose the function of the genes. But I was wondering, do you have any basic ideas or pre-consent about whether the genes variation itself and the element from structure variations, no matter insert in the stock codon or in the gene structures, which genes from genes and from gene side and from large structure variant side, which could be more affect to the phenotypes, not only the tolerance or the any phenotype.
Oh, well, honestly, I cannot guess.
Okay, it's open question.
Yes.
Yeah, okay, we will see. Yeah, we can.
Just one comment. I mean, beside where it inserted, and also what is doing, and because, for instance, I'm curious to see what will happen with methylation spillover. Let's say the TE quite likely will be methylated, but if the methylation is not limited to the element, but for whatever reason can move towards the nearby gene, that can have some effects. But all of these kind of things have to be.
Yes, it's really important because as to genes itself is already pretty much studies and the technology has focused on that a lot. But for this part, it's still a lot of blanks now. Yeah, it's good and worth to do that. So there are a lot of hands up there.
Yes, and I have another question here in chat. Hajime, how long is the time scale of the stress response? Is that happening in the next generation or needs more time? It can be pretty fast, indeed. Yes, so it could be even a matter of generation if the stress is effective in triggering the reaction. Yes, indeed. Oh, hi, Rod. Nice to see you.
Andrea, you have a question from Camilla.
Oh, and
it looks like she's waiting to ask a question. So.
Oh, okay. Go ahead. Sorry, because I cannot. I am trying to following things here, but okay. Sorry, Camille. Please.
No worries. No worries. I'm very patient. Thank you so much. It was a beautiful talk, and my question was related actually to your answer. To be curious how to replace the correlation with causation, which was one thing, because I'm a statistician. I'm not a plant scientist. I'm not working in bioscience. I'm a statistician. And I would suggest maybe something worth to think about. It's just like an open thing about Bayesian analysis, which actually uses conditioning on the given data, and it gives the causation relation. because the correlation sometimes can be misleading. Like we can have a correlation between production of energy in Australia and production of chocolate in Switzerland.
I mean,
but what is yeah.
I'm well aware of this, and you correctly pointed that out. And so definitely I see two things that we should do. One is what you suggest. You were suggesting Bayesian analysis, but more generally, I mean a proper ad hoc devised statistical analysis of this data. But then, even if I am a bioinformatician, I still think that at one point we will have to go back to the wet lab and see what happened there, inserting or removing a certain candidate TE nearby a gene, and see if that is the real agent of what we see and things like that. But no, I am aware that the statistical analysis will not be trivial. Also because, I mean, we are going to produce a fair amount of
challenge,
data.
there is opportunity.
Sorry?
In every challenge, there is opportunity.
Oh, yes, indeed. As I said, I am excited about that.
Okay, and the second question, to not take too much time, was about the relevant study in the human genes, because it's so exciting to hear about that the stress affects the genes and therefore the response of the plant. So I'm really interested in bridging the science and spirituality, and I think that epigenetics is one of the domain which is actually doing something towards this more holistically than anyone can think, actually, that the environment actually affects the cells and therefore the gene response, and that gives the body response, and therefore
I got your point, and I will try.
And then, like, the point was just to have like a brainstorming idea from your experience with plants, and if you're aware of any studies in this.
Yes, that was part of my answer, and if you contact me, I can send you a nice paper where they were dealing about the rewiring, I mean, the variability of and the insertional polymorphism of retroelement in the cells of the human brain, okay, during development and later on. And basically, they were pointing out how stress can trigger element activity, and insertional polymorphism can also have an effect of on behavior, okay? And again, it was really speculative, but it was quite fascinating. But I can send you that paper, and then we can discuss.
Thank you so much.
No, you're welcome. Just
you.
contact me. I mean,
yes.
I know where it is.
Andrea, you have another question from Salim.
Oh, okay.
Hi, Andrea. Can you hear me?
Yes, I can.
Lovely, beautiful talk. Thanks very much for that.
Oh, hi.
I actually know very little about TEs, so I'm going to ask a question, but it's not going to be biological, and do pardon it if it's a bit ignorant. In the beginning, you explained that the removal process, and you compared Norway spruce to rice, and then you said there are mechanisms that would basically they're fast in rice and slow in spruce.
Yes.
And then when you compared the African rice to Oryza sativa,
yes,
you described how you would look at basically when TEs basically were before the divergence or after divergence.
Yes.
How can you tell if for example a TE was there before divergence and then it kind of it basically was removed after the divergence using some sort of removal mechanism?
Oh, well,
how can you differentiate that?
Yes, I mean, that was inserted before is, I can tell you that because I see that very same element in the very same position on the two genomes. And so there are two possibilities, or we have had two different insertions in the very same position in two different genomes, but that would be quite unlikely. Or the element was inserted before the two species separated and was retained there. Okay.
Mm.
Then,
yeah, but how can you tell whether a TE has been inserted after the divergence? Because it could have been, inserted early and then removed afterwards.
Actually, I didn't spend the time to describe, because there is also the alternative position that what we see is not an insertion, but a removal in the other species. I mean, it is present in one, absent in the other, has been inserted in one, or has been removed in the other. You can tell that looking at some signatures, because when the element is removed, it left behind some signature, and so if you inspect the sequence, you can tell that. But I mean, I keep it simple, admittedly, yes, and I was oversimplifying, and also the most
thanks for that. Obviously, I'm grateful to that.
You're right. And the most effective way to sort out those kind of things actually is in I was describing that in the slide that I skipped, and it is having more than 2 individuals, at least 3 or more, even better, because at that point you can polarize the events and sort out real insertions from real deletions. Okay. But again, you know, it's nice that you spotted that, because that means that you have paid a lot of attention.
It was a great talk. I gotta applaud you, man. That was a beautiful talk. Thanks very much.
Thank you.
Andrea, you have a question from Hajime in the chat box.
Yeah, no, I think I replied.
Oh, yes.
Did you answer that?
Yes,
it's already answered. Yeah, thank you.
There is another, I think, from Robert just before that. He asked the second question, I think. I don't know whether you've seen it or not.
Okay.
Oh, yeah, okay. The chat above Hajime's.
Okay. Oh, yes, the question was, Robert was driven, and that. Do you also see karyotypic changes after T expansion events like chromosome fusions? Well, actually, I never checked them, but no, I cannot comment on that, honestly. Not in my experience, at least.
Okay, Andrea, there's I guess no more questions, but I apologize for not introducing you. I had a meeting with 2 Saudi princesses at 2 o'clock in the morning, my time, and I took a little nap. And anyway, but for everybody who's still on, I would like to introduce Andrea just for a second, just to say that we've worked together for many, many years, and he's a tremendous resource for us, a real professional bioinformatician in transposable element biology. So I would definitely tap into his skills and knowledge at KAUST, because he's invaluable for advancing our work in rice and many other genomes. And particularly coming up in the native genome project that we're working on. So it's anyway. Thank you, Andrea. Thank you for joining me on this adventure at KAUST.
No, thanks to you.
Yeah.
Really
so I think with that, maybe we end the talk. And. Please join me in thanking Andrea for a wonderful talk today. And everybody, you have a wonderful, wonderful rest of your weekend.